Hi, hello, and welcome to Dr. Vinita's bio classes. So we are continuing with our first chapter on sexual reproduction in flowering plants, and today's topic is post-fertilization, structures and events. In the previous class, we had seen about the embryo being formed from the zygote and the endosperm being formed from the triple fusion. The development of the endosperm, the development of the embryo, the maturation of ovules to seeds and ovary into fruit, all these collectively is known as post-fertilization events. That is, the endosperm development, embryo development, the seeds will be developed from the ovules, the fruit will be developed from the ovary, and all these events are collectively known as post-fertilization events. Okay, so we'll be going through each and every structures involved in the post-fertilization events. So first structure is the endosperm. This endosperm development happens before the development of embryo. And what would be the reason for that? The primary endosperm cell, which was formed as a result of triple fusion, it undergoes division over and over and it forms a primary endosperm tissue. And as discussed in the previous chapter, what would be the ploidy of the endosperm tissue? It is triploid, right? We saw that the three nucleus, that is the two polar nuclei and one nuclei from the male gamete, all these three are haploid. So three haploid nucleus, they had fused to form the primary endosperm cell. And so its ploidy is triploid, right? And what is the function of this triploid endosperm tissue? These tissues are filled with reserve food materials. Okay, so endosperm tissue, it has reserve food. So it has reserve food in it, and so it provides nourishment to the developing embryo. So for the embryo, for its proper development, it requires this nourishing tissue, which is the endosperm tissue. So that's the reason why the endosperm tissue is developed before the embryo development. Okay. And saying about this nucleus in the primary endosperm tissue, that is the primary endosperm nucleus, it undergoes free division. Okay, it undergoes repeated division, and as a result of that, several free nuclei will be formed, and this will be called as free nuclear endosperm. Okay, that is the primary endosperm nucleus, it undergoes repeated division to form free nuclear endosperm, and this is followed by cell wall formation. So this nucleus undergoes repeated division and later on the cell wall formation occurs. And how many nuclear division occurs in the pen or the primary endosperm nucleus varies from species to species. For example, in the case of coconut, you know that coconut has water inside it, right? And that water is actually the free nuclear endosperm. Okay, so that water in the coconut is the free nuclear endosperm, and outer to this water is the endosperm. The white kernel in the coconut is actually the endosperm, and this kernel, it gives nourishment to the developing embryo. Okay, and there are cases when this endosperm is completely consumed by the embryo even before the seed maturation. Okay, for example, the groundnut, the beans and the peas, these are examples wherein the developing embryo consumes the endosperm much before the formation of the seeds. And what they do is that they store these endosperm in their own seeds as cotyledons. Okay, you have studied about monocotyledons and dicotyledons. right? So these cotyledons, they serve the purpose of endosperm in several seeds. And there are certain other examples such as the castor and the coconut, wherein the endosperm is preserved, and this endosperm, it will be used by the germinating seed. Okay, so in castor and coconut, the endosperm will be preserved and it remains during seed maturation, and it is used up during seed germination. So endosperm, it can either be consumed completely by the developing embryo, or it can be preserved. It can be preserved in the mature seeds and it can be used during germination. Okay, so both these conditions are also present. And in the case where the endosperm is completely consumed by the embryo, the food reserve will be stored in the cotyledons. So these two conditions are also prevalent in the case of endosperms. So the next structure in the post-fertilization events is embryo. We already saw that the embryo is developed at the micropylar end, right? The male gamete, which enters through the filiform apparatus in the synergid cells through the micropylar end, it fuses with the egg cell and forms the embryo, right? And this embryo, it develops only when some amount of endosperm is formed. Because we already saw that the endosperm, it gives nutrition to the embryo. So if the embryo is formed before the formation of endosperm, you know what would be the fate of it, right? So that doesn't happen. It's always after the formation of endosperm that the embryo is developed. At least some amount of endosperm has to be present for the embryo development. There is a term here which is called as embryogeny. Embryogeny simply means the early stages of embryo development. Okay, the early stages of embryo development is known as embryogeny. And this embryogeny is similar in both monocotyledons as well as dicotyledons. The embryogeny, that is the formation or the early stages of the embryo development, is same or similar for both monocotyledons and dicotyledons. The zygote, it gives rise to proembryo. This proembryo, which is formed from the zygote, it then becomes a globular structure, and then it becomes a heart-shaped structure, and which is followed by the formation of the mature embryo. You can see it on the figure. Okay, can you see it? The zygote, it gives rise to the proembryo. And then you can see a globular structure, develops into a globular structure, then it becomes a heart-shaped structure, and finally the mature embryo is formed, right? So if we say about a typical dicot embryo, dicot means dicotyledonous. So if we say about a typical dicot embryo, you can see that the embryo consists of an embryo axis. Can you see the embryo axis? Okay, it has an embryo axis and it has two cotyledons. Because it's a dicot, it will have two cotyledons. Okay, so the dicot embryo, it has an embryo axis and two cotyledons. Okay, and the embryonal axis, you can see that the embryonal axis, which is above the level of the cotyledons, you can see the cotyledons, and above the level of the cotyledons, the embryonal axis, which is above this level of the cotyledons, it is called as the epicotyle. Okay, you can see the epicotyle, which is above the level of the cotyledons. Okay, and this terminates with the plumule. You can see the plumule, right? Plumule is otherwise known as the stem tip. Okay. So basically it means that the structure which grows towards the top is known as the epicotyle, and it terminates in the stem tip, which is otherwise known as the plumule. Okay, And you can again see that the cylindrical portion which is below the level of this cotyledons. See, there is a structure which is shown to come below the level of these cotyledons, right? And that is marked as hypocotyle. Hypo means less or below. Okay, the word meaning is below. The cylindrical portion which is seen below the cotyledons, which is seen below the level of these cotyledons, is known as hypocotyle. And this hypocotyle, it terminates in the radicle or the root tip. Okay, so the end of this hypocotyle is known as the radicle or the root tip. That is the portion of the cotyledon which goes towards the ground is known as the root tip. The structure is known as hypocotyle and the end portion is known as the root tip. And this root tip, it will be covered by a root cap. Okay, can you appreciate all these structures? Okay, so that's about the embryo of a dicotyledonous plant. So we saw that the dicot embryo will have two cotyledons, right? And the monocotyledonous or the monocot embryo, it will have only one cotyledon. And the example of a monocot embryo is the grass family. Okay, grass. You know that grass is monocot. Again, you can see in the picture that the cotyledons of the grass family, it is known as the scutellum. Okay, you can see the scutellum. It is only one cotyledon and it is called as the scutellum. And you can also see that this scutellum is towards one side of the embryonal axis. Can you see it? It is towards only one side of the embryonal axis. And towards its lower end, this embryonal axis, it has a radicle. The lower end of the embryonal axis, it becomes the radicle or the root tip and it has a root cap. Okay, and this portion, that is the radicle and the root cap, it has some undifferentiated sheath and that is known as the coleorhiza. This undifferentiated sheath is called the coleorhiza. And again you can see a structure here also called as the epicotyle, which is the structure or the part or the portion of the embryonal axis which is above the level of the scutellum. Okay, so the scutellum you can see, and above the level of the scutellum, that portion is known as the epicotyle. We had seen the epicotyle in the dicot embryo as well. So here also the structure which is above the scutellum is known as the epicotyle. Okay, this epicotyle, it has a shoot apex. Okay, that is a structure similar to the stem tip in the dicot. Here also the epicotyle has the shoot apex and there will be few leaf primordia. Okay, so that's the same for both dicot as well as monocot. At the shoot apex or the stem tip, there will be leaf primordia. We had already seen about the what is meant by primordia. So here in the epicotyle, there is the shoot apex and it has few leaf primordia, okay, which will be enclosed in a structure called as the coleoptile. Okay, so here another term is coleoptile. So the part which goes towards the earth is called the coleorhiza, and the foliar part or the structure which goes above this scutellum, it is called as the coleoptile. Okay, so there is coleoptile which grows towards the top and the coleorhiza which grows towards the bottom. So that's about the embryo. So we have covered the two structures in the post-fertilization events, that is the endosperm and the embryo. The third structure is the seeds, which will be covered in the next video. Thank you.